Starting with, I'm going to use flower colour because it's easy. Basically, Mendel was a botanist. He was also a monk, which meant he had a lot of time up his sleeve, which is good. A lot of the best science was done by monks and people like that because they had time, a lot of reflective time to think and to do really good experiments. One of the things Mendel noticed was that with peas, there was quite a lot of variety. And one of those things that had variety was flower colour. So we'll say, for argument's sake, most flowers were red and some flowers were white. So what Mendel wanted to know is why were there none that were pink? And he wanted to see if the flower colour was inherited or due to, say, conditions in the soil or to do with the atmosphere. And so he designed a series of experiments which are pretty awesome actually, especially for the time. And what he did is he bred red flowers until he was pretty sure that they would only give red flowered offspring. So he basically created a group of pea plants that were homozygous, we use that word now, homozygous dominant. Then he had some white flowers and did the same thing, but actually what he noticed is when you crossed white flowered peas with white flowered peas, you always got white flowered peas. There was no other option. So that was a bit easier to make. Nowadays, we'd say that these are, for example, big R, so they're purebred or homozygous dominant, and these would be little r, for example. Little r, they're purebred or homozygous, in this case recessive. He didn't know that though. What he did is he bred red ones with white ones, and what he did is quite simple. He got a paintbrush basically, and he took the pollen from red flowers and put them into white flowers and vice versa. But he didn't let red go with red or white go with white. It was just red with white. And what he noticed is that all of the offspring, regardless of how many he had, were red. Red flowers, not red plants of course, had red flowers. And none of them had pink, none of them had white. So there's no such thing as a pink flower. They were red or they were white. And if he crossed these two together, then all he got was red. So then the next part of his experiment was, what happens if I cross these two together? And what he found is that about a quarter of them had white flowers when he did this. So when he crossed these two, he found that we got red. I'm not going to do the numbers because I get sick of drawing flowers. He had some red ones, and he had some white ones. So what he decided, or what he concluded, was that these ones must be carrying the genetic information for white flowers, but not expressing it. So coined the term basically that it was a recessive, or hidden, or carried characteristic or trait. All right. What we know now, of course, is that these were big R, little r, which means they were what we'd call heterozygous. They had the allele for red and the allele for white. Because the allele for red is present, the white allele doesn't get turned into a protein, so it isn't coded for, it doesn't do anything. Only the red one gets turned into a protein, so the red colour. But what it does allow them to do when crossed with each other is potentially have some white offspring. And with a big enough population, we get 3 of those for every 1 of those, a 3-to-1 ratio. So, as genetics goes on, we can start using some tools to help us out. We can check this by doing gene sequencing and all that. But even before all those awesome techniques were invented, people understood how this inheritance worked. And they did that basically by a lot of experiments like this on different characteristics which are controlled by one locus or one place on a homologous pair of chromosomes. So the alleles are found at the same place, and you either have, in this case, big R or little r at that point. Those different alleles have got different base sequences we now know, which is why they code for, in this case, different colours. They're also pygmy, so very small pea plants compared to normal size. That was also controlled by one gene. There's no in between fully grown. They're either pygmy or they're normal size. And there was a rough seed coat or a smooth seed coat. That was another one that Mendel experimented with. What he found is you could actually use a really simple, almost noughts and crosses type diagram to show how this probability works. And we call these a Punnett square. Now, our understanding of meiosis helps us know this as well. We know that when you have meiosis, you start with your two alleles, so one on each of the homologous pair. One goes to two of the gametes, and the other one goes to the other two gametes. So if I was big R, then all of my gametes are big R. They're just haploid, so just one big R. What I'm going to do here is I'm going to show, first of all, why all of the offspring were red. So we've got big R, and the little r. These boxes here represent the gametes. Now I know that for every one meiosis event you get 4 gametes, so it's just a ratio. Okay. So little r gives us 50% little r and 50% little r, so all of them are little r. Same thing here for the big R one. Alright? When these cross, this is just showing a probability of what the offspring's genotypes will be, so the allele combinations. And so we get big R, little r in all of them. So they're all heterozygous. This Punnett square shows us why all of the offspring will be red, because they all have the dominant allele, which means they're all going to express the dominant phenotype. Now I'm going to show you why there was roughly a 3-to-1 ratio here. And when Mendel did smaller sample sizes, he noticed this ratio didn't work. And he actually was a bit naughty, if you want to be true for science. He threw out the results that didn't fit the 3-to-1 ratio when he had a small population size. But whenever he had a large population size, he got close enough to it. There's more things happening than this. Luckily, you don't have to know about them this year. But basically, this works really well for a large population. So, in this one here, it's basically the offspring being bred together. Not such a dodgy thing to do with plants. Probably wouldn't do it too much with animals. What we do here is we now know that all of the offspring have the same genotype. Big R, little r. So we like that. Which means half the gametes for this one are big R, half the gametes for this one are little r. Half the gametes for this one are big R, half of them are little r. Then when we do the cross, well, there is a 1 in 4 chance that these two will meet. That this, for example, this is plant. This pollen will meet this ovum. So there's a 1 in 4 chance that big R. Or there's a 1 in 4 chance that this pollen meets this ovum. So it'll be big R, little r. And so on. Each of these boxes is a 1 in 4 chance of a particular pollen fertilising a particular ovum. It's not saying that you get 1 of those, 2 of those, and 1 of those. It's all probability. We know that in a little more depth now. So what we'd expect here is that one of them will have the recessive phenotype, because only one of them has got recessive alleles only. And in a large enough population, there's a 3 in 4 chance of the dominant phenotype being expressed. But actually, there's only a 1 in 4 chance of there being a homozygous dominant offspring. We can then look at pedigrees or family trees to see how this works. And one of the best ways to look at it, I'm just going to use colours again. If I had 2 red flowered parents crossed, and regardless of the ratio, I've got a mixture of red and white flowered offspring, I know that both of these parents have to be heterozygous. If this was the recessive allele, you couldn't get the white ones, because they don't carry the recessive allele. If they were homozygous, you couldn't get white because they don't carry the recessive allele. So if in a pedigree chart, a family tree, you see two things with the same phenotype giving a mixture of phenotypes, or even the different phenotype if it's only one child, then you know that the two parents must be heterozygous. They must be, unless there's a mutation or something happening there, but they won't do that to you at this level. Okay, so these ones have to be. These ones have to be little r, but you have no idea what these three are, because they could be heterozygous or homozygous according to that Punnett square. So basically, in a nutshell, we've gone from Mendel doing some observations, lots of them, to an understanding of how alleles work, and then looking at family trees or pedigrees to be able to work out what those alleles would be.